Okay, we are starting this course with Chapter 1, kind of an intro course, kind of a neat chapter. I'm going to probably go into a little bit more detail than I really need to, than you need to know. Um, Introduction to the topic, who are the most important people. Most of the people I'm not even going to really get into until we see them in later chapters. Nothing really very difficult in this chapter. Okay, so the history. Lots of important people here. I'm going to talk about some of the key concepts in microbiology that we still find important today. Okay, so let's go back into the 1600s. Microbiology really didn't even start as a field of biology until the 1800s. We'll get to that in a minute. But there were some important people even before that time. And you're going to hear about this guy again because we're going to talk about him in the chapter on microscopy. This is Antoni van Leeuwenhoek. You see his name spelled, gosh, I've seen it spelled probably 25 different ways. Sometimes it's written as Anton or Anthony. It just depends on what country you're from. He was a Dutch linen merchant. He was wealthy, lived in Delft, Holland, had a shop where he sold linens, cloths. And he developed a microscope, a handheld microscope, you see it in the picture there, in order for him to be able to see the quality of the fibers, that is, how many fibers crisscrossed in, for example, a square centimeter or square inch. The higher the thread count, the higher the quality of the cloth. And so he actually ground his own lenses. Very innovative. Obviously, you get kind of bored looking at cloth all the time, so he expanded his use of his little handheld microscope, which only had one lens, by the way, as opposed to your microscopes, or the ones you know about that have two lenses, one in the objective that's on a rotating nose piece, and one that is in the ocular. This only had one lens, and again, I'll talk more about the setup of the microscope in the later chapter. I'm not going to get into it, but I will show you where the lens is. It is right there, and here it is from the side. You can see a lens right there, and your object would get put right there. These were small, probably 4 inches or so. They are considered simple scopes, almost more like a magnifying glass of a sort. But they magnified almost to the point of about somewhere around 400 magnification, which is actually really very good. That's comparable to a high power lens on your scopes. He made over 500 of these. He wrote everything down in extensive journals. He wrote lots of letters, got inducted to the Royal Society of London. That was like the premier scientific society of the 1600s. He corresponded with them for 50 years. And in 1676, he had his first letter with drawings of organisms that he found in water, in pond water. He called them animalcules. So he is credited with being the first one to see, and to record anyway, microorganisms, bacteria, protozoa. He saw algae, things like that. He also looked at other things, like he's the first one to identify sperm and lots of other things too, muscle cells and such like that. But anyway, that's his big contribution. Okay, so the organisms we're going to be covering this semester, again, we're going to be talking more about this in later chapters. Let's jump over to another guy. Again, you see him called Carol Linnaeus, Carl Linnaeus, Carolus Linnaeus. He is the father of taxonomy. He did most of his work in the 1700s. He is the first one to come up with a kingdom system. The first two kingdoms were plant and animal. He also created a third system, a third kingdom for the protozoa and little microscopic things that had been recently discovered in the latter 1600s by Leeuwenhoek, and he called that kingdom of the protists Infusoria, which never really caught on. He also came up with the binomial nomenclature that we use, the genus and species names of organisms. So, anyway, just to lay out the things that we're going to be looking at this semester, we're going to be looking at, our main focus is going to be on bacteria, which are prokaryotic organisms. And we're going to be looking at two groups, eubacteria, which are what are called the true bacteria. Eu means true. And the archaea, which are another variation of bacteria. We're going to be looking at fungi a little bit, the protozoa, the algae, and we'll spend a little bit of time on worms, which are the helminths. And you can see some different examples here, just to point some of them out. I've got a prokaryotic picture here. By the way, these are not drawn to scale, so these bacteria look really large compared to something like an amoeba, which is this guy right below them, but amoebas are way bigger. So anyway, the fungi, I've got a couple of examples here and here. The moles on the left, yeast on the right. The protozoa, I've got a couple. I've got a ciliate protozoan, and I've got an amoeba right there. For the algae, I have a single-celled alga right there, and I have a multicellular kelp seaweed right there, and then I got a couple of worms right here. So those are some of the things you're going to be looking at this semester, and we'll be talking about in lecture. Now, there's one thing that I have not mentioned. Perhaps you may have already picked up on it. You're thinking, wait a minute, what happened to the viruses? Well, the viruses are not categorized along with any other organisms. This is a virus that you're looking at on the picture, a diagram of one. Viruses are not considered to be alive. They do not fit the criteria of life, of cells. They have no cell structure. We'll talk more about that when we get into the viruses. So they're not categorized. They're not in the same kingdom as the bacteria. They're different from plants. They have no kingdom that they are in. Totally different setup. Okay, so let's go to another important topic, which was spontaneous generation, how it was disproved. You've covered this in general biology. Maybe, maybe not. You found out that Aristotle was a proponent, actually one of the creators of spontaneous generation, which basically said that life comes from non-life. So in other words, you leave, I don't know, food around, meat around, and eventually you start to see maggots growing in it, and they become flies. So it looks like things came out of the meat, that flies came from meat. Life came from non-life. Obviously, he was right about a lot of things, but he wasn't right about spontaneous generation. So you see three people listed here that are important in the disproval of spontaneous generation. The drawing that you see up at the top with the meat is the experiment set up by Francisco Redi. He was an Italian physicist and cleric, and basically he set up containers with raw meat. Over on the left you see the control, which was open to the outside where the flies could come in and lay their eggs, which then turned into maggots, and then they pupated, and then when the pupa opened up, adult flies came out. Then he set up another experiment where he put cork into the mouth, and the third one where he put cheesecloth over the container. And in the last two, obviously, flies couldn't get there to lay their eggs, and the meat never had flies come out of it. This was the 1600s, and people just weren't ready to accept this change in ideas, because up until this time, people thought spontaneous generation was true. Now, it had been kind of gnawed at around the edges, so in a lot of circles, people really didn't believe this anymore. It was thought that frogs came out of um, the mud, bees came from honey, um, just stupid stuff like that. The work done by Reddy and then by Spallanzani kind of made people think about whether this was true or not. But the problem then, in the latter 1600s and early 1700s, when microorganisms were identified, people really didn't have another explanation for where microorganisms came from. In the case of a fly, if you watch a fly behavior, they land, they lay eggs, which are very small, you can't see them, but eventually you would see maggots, which would turn into pupa, which would turn into flies. So if you actually just watch them for a couple of weeks, you could see the whole transition of the animal from egg all the way to fly. But you can't do that with bacteria. And so people kind of reverted back to this dumb idea of spontaneous generation. So in the latter 1700s, Lazzaro Spallanzani and other Italian cleric scientists, important for a lot of different things, by the way, jumped into this phrase spontaneous generation disapproval. His idea was that microorganisms come from the air, and boiling will kill them. So he set up 4 flasks, like a Erlenmeyer flask. One was open, had a fluid, a nutrient material inside of it. One was sealed with something on it. Another was boiled, but it was left open, top open. And the 4th one, the material was boiled, and then it was sealed. And he found that in the containers 1 and 2, which were either left open or sealed, but remember in flasks 1 and 2, no boiling was done of the fluid solution. There were already bacteria in there before he even put it into the flask. And you'll notice that microorganisms were found. Flask 3, the material is boiled, but it was left open to the air, so things got in there from the air and started growing the solution, found microorganisms. Flask 4, not. So this supported his hypothesis that microorganisms come from the air and boiling kills them. But again, even though Spallanzani was important in a lot of circles, he did some work on echolocation, he artificially inseminated dogs, he had a lot of clout. By the way, it's a picture of him over there. He didn't have a strong enough reputation to carry this, so it falls to Louis Pasteur, who we're going to talk about now, in the latter 1800s, actually mid-1800s, when he started his work on disproving spontaneous generation. He's the one that gets all the big credit. Okay, so how did he become famous in the first place? Because he was already famous before he jumped into spontaneous generation disproval. He was a chemist. In fact, he was a biochemist. He was not a microbiologist. That came along later. He's in France. He lived from 1822 to about 1895. And he was working with spoilage of wines. He was asked by the wine industry to get involved in this. So his questions were, was there something in the air or something else chemically about wine that caused a change in the wine, or change in the grape juice in its production of alcohol? Up till this time, the early 1800s, people just thought that alcohol was like a decay product of grape juice or whatever you made it out of. They really didn't see that yeast or anything living was really involved in fermentation. Louis Pasteur is the first one to really prove that. And of course, his second thing was, where do these microorganisms come from? And that takes us back to air. And that then set up the stage for him to jump into spontaneous generation. We'll talk more about his contributions in wine later. So if you look at these flasks on the picture over to the right, you see some different flasks with solutions. This was a nutrient solution that he made up. It boiled it. Started out with boiled material. The necks of these flasks are open to the air. Remember that he wanted to show that these things came from air, that it was air that was the major contaminating material that contributed to microorganisms. So he set up all sorts of flasks with these openings, and in some of the open flasks, he found that eventually, over a few days, something started growing in his flask. Now remember, he could use a microscope, he could see them. Besides, the broth, which started out kind of a dark, clear tea type of material, became cloudy as the microorganisms grew. So you could see it microscopically and you could see it to the naked eye. But when he came up with these two shapes down here, things were a little bit different. The shape, which is called a swan neck or gooseneck flask, allowed air to get in, but the microorganisms settled in the deep neck areas right over here, and they could not get up the neck against gravity into the flask, because microorganisms can't fly through the air. It's air wafting around and moving that carries them around. So he did a variety of experiments looking at different kinds of air, urban, rural, different altitudes, amassing a whole bunch of data on how different types of air had different contamination rates and so on, and eventually disproved spontaneous generation. He could keep these flasks at the bottom sterile for years and years and years. In fact, there's some still in the Pasteur Institute. So, let's move to disease. Here's some thoughts about what people thought was causing disease. The oldest, of course, is that it was punishment. You did something wrong, and God or gods, if you were Roman or Greek, you believed in lots of gods, punished you. Or we had scapegoats like lepers, Jews. Plague was blamed on gypsies, also dogs. Sometimes they were animals, sometimes they were humans. There were a lot of recordings of infectious diseases, herpes mentioned, also in Romeo and Juliet in the 1500s. Writings about vaccination for smallpox, that's what variolation is. Documents that talked about dying rats being an omen of plague, and of course they were dying of plague carried by fleas carrying the bacterium. I want to talk about Hippocrates here. He's the father of medicine, 400 BC, Greek, really important. Wrote something called On Air, Waters, and Places, and in it he talks about poisonous vapors or smells called miasms. And that eventually led to the idea of the miasmatic theory of disease, which was basically bad smells cause disease. And a good example of this, of a disease which fits this, is malaria. And the word malaria means bad air. Now, it was known that if you lived around slow-moving pond water, river water, that your possibility of getting malaria was much higher than other people. And it also smelled bad, so they thought that it was the smell of kind of a rotten egg, hydrogen sulfide type of smell that you get from the sediments in slow-moving water and muds and things. In reality, obviously, mosquitoes would rather breed in slow-moving water than fast rivers, and of course they carry the protozoan that causes malaria, but those two pieces of information were, they didn't know about it. In the 13th century, Sir Francis Bacon theorized about invisible creatures, so he wasn't too far off. And then in the 1500s, we're going to talk more about Fracastora put this idea a little bit even more out into the public for them to get an idea of. Okay, so let's go to the most important people. Germ theory of disease says that microorganisms cause infectious diseases. They could be transmitted any number of ways, but they are the etiologic agents, infectious agents of a disease. There are three people here. Of course, you recognize Pasteur's name there. So I've already mentioned Fracastora. Again, we go back to an Italian. He's an Italian doctor. He's an Italian cleric. He's a poet. He writes an important piece of literature called Syphilis. It was about a shepherd who made the god Apollo mad because he was dallying around with a girl when he shouldn't have. And Apollo put sores on his body, and he was basically describing the secondary syphilitic rash. And as I mentioned, the name of the shepherd was Syphilis. It's a multi-page poem. Fracastor was also a doctor. He wrote an important publication called De Contagione, or Contagion, about transfer of infectious diseases. He didn't call them bacteria. He called them seeds, invisible seeds. And in fact, he is responsible for the seminaria theory of disease. Semen means seed, and that proposal was Fracastora again in the mid-1500s. Okay, so let's go back to the miasmatic theory. So in the latter 1700s and then early 1800s, things were really taken off in Europe. Industrial Revolution, things becoming better. But there were some things that were not very good, and that was a lot of pollution in the air, a lot of factories, things like that, and you could see the effect on the air and people's health. And so people embraced that miasmatic theory, again, the idea that bad air causes disease. So it really took hold quite a bit in the 1800s. Remember, it was wrong, obviously. So let's jump into the 1800s, and again, we're in the mid-ish 1860s, 70s, and we're at Louis Pasteur. Now, he's already become famous for his work in the wine industry, and he's also become famous for something else. He was asked by the silkworm industry to figure out what was going on with the silkworms, the larvae that they use to make silk. They make a silk protein, and I don't know how, but the silk protein is removed and turned into silk clothes. And these little guys, the little caterpillars that make the silk protein, get a couple of parasitic diseases, and Louis Pasteur was asked to get involved in this and figure out what was going on. He already had a reputation for his work in the wine industry and his development of pasteurization and stuff like that. So he figured out that it was an infectious disease and went through various steps of how you could reduce infections in your silkworm caterpillars, which if you were a silkworm caterpillar wrangler, owner, this would be devastating monetarily to you. He then kind of changed his focus and moved into spoilage of milk, which isn't a big jump from spoilage of wine that he worked on earlier, and came up with the idea of, well, if microorganisms can spoil wine, can spoil milk, can cause infections in caterpillars, then there's not a big leap that you have to take to understand that the same thing can happen in humans. And so he starts working on his own germ theory of disease. He's using chicken cholera in chickens, not really related to cholera in humans. Researching microorganisms, how it's transmitted from chicken to chicken, and so on, and develops his own germ theory of disease. And then, of course, is working on rabies vaccines in the latter 1800s and so on. But the one that gets all the glory is Robert Koch, and that's the picture you're looking at over here. And we're going to look at what his big contribution was in just a minute. He's sometimes considered the counterpart, the German counterpart of Louis Pasteur. Louis Pasteur was about 15 or so years older, and Facts, data really don't show that they ever corresponded. They didn't run in similar circles. France and Germany were at war with each other in the 1870s. But anyway, they are the French and the German counterparts. Okay, so Koch and his germ theory. His germ theory was called Koch's postulates, and he really didn't even come up with them. He was under the tutelage of Friedrich Henle, as in Henle's loop, if you've had A&P, kidney. Robert Koch kind of refined them, and he published them. That's his big claim to fame. He published them. He's the first one to get them down on paper. And what they do is they definitively link an infectious disease to a specific agent. For example, Vibrio cholera, one of the organisms that he used, that he became famous for figuring out. The bacterium Vibrio cholera causes cholera. As a result of his work, it was absolutely known that the agent of cholera was that bacterium. Cholera was not caused by anything else other than Vibrio cholera. Koch also figured out that Mycobacterium, I'm not going to spell it out, but I'll just put TB for tuberculosis, causes tuberculosis. He figured out that Bacillus anthracis, another bacterium, causes anthrax. Those were the three biggie organisms and diseases that he figured out. Okay. So, how did he do this? He did this in 4 steps. These are the postulates. The first postulate says the organism that you determine to be the cause must be present in every case of the disease. So in other words, if you're studying 100 cattle that have anthrax, all 100 have anthrax, you should find Bacillus anthracis in every single animal. Okay, so if you have these animals with similar symptomology, which have the same disease, you take a specimen. So, number 2, you take a specimen and you grow culture, whatever you want to call it, the organism, in a laboratory, and you run tests on it. So you can see that the organisms, the specimens, are being looked at in the microscope, being grown on agar plates, tests being run on them, blah, 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 blah. And again, if you sampled all 100 cattle, you would have the exact same results right here for all 100 specimens. Okay, 3. You have this tube now of the microorganism. It's growing in a test tube. Let's say it's Bacillus anthracis. Now, remember, nobody's ever done this before. Nobody's ever gone to the trouble to tie an organism with the disease. So they were not named. You would just call it bacterium X because it had no name. All right. So anyway, there's your organism. So in order to verify that this is the organism that causes the disease in these cattle, what if we took this culture and put it into healthy animals, like more cattle? Cattle are kind of bad test animals. Probably want to use mice or rats. And of course, you would also want to recreate the symptoms. You would want the same symptoms. I'm going to put SX, S for symptoms. The same symptoms as the original 100 cattle. They can't be different symptoms. 4. You take your newly diseased animals. You take more specimens, re-isolate the organism. It checks out to be the exact same organism as in step 2, and there are the 4 postulates of Koch. So this opened up a whole new world for people to figure out what was causing what. I'll show you an example in a minute. Okay, some other people that are involved here. Many of them had to do with Koch, by the way. Koch, remember, in that he's the first one to ever do a methodical set of tests to link diseases and agents that cause it, had nothing to work with. I mean, he didn't have media, no bacteriological media. He had no tools. Um, the microscope was around. It's not like he had to invent that. But he had no procedures, nothing written down. He had to invent everything to do this. So he's sometimes called the father of clinical microbiology, because he had to invent ways by which you could grow microorganisms, most of which are still used every day a gazillion times in labs, both teaching and clinical labs. Okay, another guy that will come up later, we're going to be talking a lot about, is Christian Graham, who developed the major staining technique we use for bacteria. This is a Graham stain of cells that you see on the right. That big pink guy is an epithelial cell from the mouth, and the little purple guys are bacteria from the mouth. Hess, which you see in a picture down below with his wife Fanny, was the first one to come up with a way of making bacteriological media, and that was then used by Koch. He actually worked in Koch's laboratory. It was a solidifying material called agar, isolated from the walls of certain types of red algae. And Koch saw this material brought to lab for his lunch, because people added sweeteners and fruit and things like that to this agar material, kind of like a jello, but not the same, and thought that it would be very handy for growing bacteria on a solid surface. So that was the beginning of bacteriological media. Julius Petrie, who also worked in Koch's lab, came up with a glass dish, I'll call it, to grow bacteria in, to put the media in, to solidify, and then you could inoculate the bacteria on. That's a Petri dish. You'll see them in lab. And of course, Koch is down there again, related to aseptic technique, techniques used in a laboratory to reduce contamination of your bacteria that you're working with, and so on. Okay, so I mentioned that the first work was done with anthrax, and Koch's publication was 1876 about anthrax. Notice that he's also up here for TB, and he's up there for cholera, as well as anthrax. And look, at year by year by year, it was a wonderful time to be a microbiologist, a budding microbiologist. There were all these diseases that had no cause that anybody had delineated, and you could just set up a lab and start working, and if you were lucky, you got a Nobel. Well, Nobel Prizes were not even given first until about 1903, 1904. Koch got one of the earliest ones. And if you were lucky, you got your name on the microorganism. For example, Theodore Escherichia ended up with his name in the genus Escherichia. An extra plus. As a result of this work of Koch and Pasteur, everything opened up in the latter 1800s. Immunology didn't really start big time until the mid-1900s, but actually it started in the latter 1700s with Edward Jenner and his vaccine, although Jenner knew nothing about immunology and didn't even know it was a virus that caused smallpox, but we'll talk about that later. Florence Nightingale and her contributions to nursing. She was very big on aseptic technique in the mid-1800s and established antiseptic procedures in hospitals so that the patients weren't as likely to get infections and die. John Snow, and who's the father of epidemiology, the study about disease in populations, and his infection control in the mid-1800s in London, working with cholera. Lister and Semmelweis, two very important people. You see their two pictures right there in the middle of the page down at the bottom. Contributions to antimicrobial technique, hand washing, things like that. And Paul Ehrlich's magic bullet, field of chemotherapy, antibiotics, introduced in the early 1900s. The time from 1860 to 1910, 50 years, called Golden Age of Microbiology, starts with the work of Pasteur and fermentation, um, goes to the death of Koch. He died after Pasteur did. That was a great time to be a new microbiologist. And as a result of their work and others, you get all these contributions you see down below. Look at differences in deaths over comparing two different centuries. If you look at 1900, I have boxed infectious diseases in the top 10. But if you look over on the right, you see heart disease, cancer, stroke have now taken the place of flu, pneumonia, TB, gastrointestinal infections, and there are still some infectious diseases, but they've moved lower down on the list. So again, you can see that we're healthier, at least from the standpoint of infectious disease. Now it's mainly diseases of lifestyle, related to lifestyle. Okay. Just in case you're thinking, gee, if I were to become a microbiologist, there's nothing for me to identify. Oh, no, no, no. There's always a new disease. You're all old enough to remember 2009, the new variant of the swine flu, H1N1, that you will be vaccinated against, believe me, somewhere down the line when you get into a hospital. And if you remember before that, in 2003, sudden acute respiratory distress syndrome caused by SARS virus. And so they just pop up, and just when you think they're under control, you find out they have ways around our control. The different areas that we're going to be going into this semester: bacteriology, rather obvious; virology, rather obvious; mycology, the study of fungi; phycology, the study of algae. Not really going to talk much about algae because they don't cause much infectious disease, but maybe a little bit. Parasitology, the study of parasites, and this includes protozoa and worms. And then we're going to switch gears, and at the end we're going to talk about host resistance, and that will be immunology. And if you've had other classes, you will find that some things come back from those other classes in micro. And if you take classes after micro, like immunology or molecular biology, you'll see some things from micro carry into those courses. And some of the contributions of microorganisms. So we'll talk a little bit about contributions in agriculture, biotechnology. We have a whole chapter on. Talk about their contributions in metabolism and diversity, and of course disease, and a little bit maybe on food. And I think I'll stop here. I'll talk more about historical importance of microorganisms in lecture, and that is it.